let's see. Tell us your favorite color, something interesting, and then show us your favorite dance move. Just messing with you. Just messing with you. Jeez.
I was just looking over here. I was like, who's watching it?
Why am I not, this is not good. There it is. Okay. Let's talk about mutations. So, here's the very simple central dogma of biology that we talked about earlier this semester. Obviously, we've gotten much more complicated this time. Mutations are heritable changes to the genetic code. We've talked about how you can pass on changes to your DNA that are not genetic mutations. That would be epigenetic, like DNA modifications, like methylation, these kinds of things. We're talking about actual changes to the DNA. So way back, even before I was born, that far back, people were studying this. I think it was back in the 20s. And there, at the time, the mentality and the thought was, if something is living in a happy, healthy environment, it's apparently well adapted for that environment. There's no need for it to change, and so it will not change. If you want to get an organism that is different, if you want to make genetic changes that are heritable, pass those on, what you need to do is change the environment. So you put the individual in an environment that's not good for it. It will then mutate and accumulate mutations until eventually natural selection identifies a version of that that's good. The classic example of this was, you guys know what the Cold War was, right? You learned about this thing. I lived through it in Calendar. It was pretty unpleasant. So the Soviet Union and the US were engaging in these economic and industrial competitions with each other. And the chief geneticist of the Soviets believed this thinking, which was actually disproved really in the 20s, but believed that if you had wheat and you exposed that wheat to cold temperatures, you would induce mutations in the wheat that would make the wheat more able to survive cold temperatures. And this is important because a lot of the world, or a lot of their Soviet area, had very cold temperatures, and it would be of value to have a wheat that could handle the colder temperatures. And so every year, Soviets would take massive amounts of wheat, and they would put it into these freezers, and they would leave it for extended periods of time, knowing that they're going to be selecting for things that can grow in cold environments that way. And what happened in the United States instead is we had a different understanding, which I guess I'll talk about what was happening in the United States. So here's the classic experiment which demonstrated how mutations actually happen. So the first is, if things accumulate in mutations in response to a stimulus, if I had a whole bunch of different tubes of things, if I had 4 different tubes of this organism, so let's say I've got a tube like this, and in here there's 1 million bacteria. If I put a poison in here, what will happen is some of these organisms will accumulate mutations that adapt the organism to survive the poison, say an antibiotic. And if I did that 4 times, I should get organisms in every single case that would come out and be resistant to that antibiotic. Because putting it in will force some here, and force some here, and force some here to develop resistance. The competing hypothesis was organisms are just living. It doesn't matter what the environment is. If you have a tube of 1 million organisms, by chance some organisms are going to accumulate mutations. And by chance some of those mutations will allow antibiotic resistance. And so you'll randomly get some that will come out of it. And maybe none of the organisms in here accumulated those mutations. And so none would become resistant. So the idea is if the environment induced mutations, if you set up a whole bunch of tubes and check for antibiotic resistance, you got to get resistance in all of those tubes. But if resistance happens just randomly, if you have organisms growing in a tube and you put them in the environment, you will have some that are already resistant because they accumulated mutations. Does that make sense? So you don't have to wait time, it's already there. So classic set of experiments basically showed, yes, there are tubes that had mutations, original mutations in them, and that those tubes made lots of colonies that were resistant. Tubes that had no resistance, all the organisms would get killed by the antibiotic. In this case, I think it was a virus is what they used. So it showed that mutations just happen randomly. So based off of this, in the United States, we realized you need to grow the organism and put it in the bad environment and select it in that environment. And things that grow better in that environment have changes in their DNA that make them so. And so we engaged in artificial selection. So in the decades that the Soviets were taking their harvest, throwing it in the freezer, and then bringing it out, planting the same garbage the next year, they were not selecting over time for things that were getting more cold resistant. But in the United States, when we were taking our wheat, we were saying in the field, this plant looks healthier than this one, than all the rest of the field. This plant has accumulated mutations that make it better to survive cold temperatures. We're going to eat all of these seeds, and we're taking these small number of seeds, and we'll grow that up. Whatever mutation happened there, we're going to propagate that, and we'll never grow these other guys again. We'll grow these guys again. So we selected after generation, and we had a very, very viable, very abundant, greater harvest per acre, more drought tolerant, more cold tolerant wheat. And probably politicians will tell you, oh, it was the nuclear arms race. Oh, it was whatever. It was capitalism and market, the free market. Probably what caused the Soviet Union to collapse is for decades, they expanded their population, their infrastructure, and their needs, but they did not improve their natural resources in their breeding lines of their crops. Whereas in the United States, we can get the highest number of bushels. It used to be if you got 20 bushels of corn from an acre, that was a lot of corn. The most recent record for number of bushels of corn from 1 acre was just recently, the new record was set at 300 bushels an acre, more than a 10-fold increase. Part of it's genetics, thank you, Monsanto. Part of it is natural selection, or artificial selection on our part.
What a funny question.
How's that sound?
What is a bushel? Like,
A bushel is a particular size or weight. I don't remember. I think it's a particular weight. Yeah, I'm guessing.
Cool. Thank you for that. Tad bit to be silly.
Okay, so let's define some terms. I hope you are familiar with these terms. But a mutation is what we would call a mutation is more correctly called a forward mutation. If you have a wild type sequence and there becomes a genetic change, in this case we get a substitution at 1 nucleotide, this is now a forward mutation. You could get another forward mutation where this mutates. What did it change from a G to an A? This changed to a C. It would be yet another forward mutation, 2 different. And the reason we call it a forward mutation is if I mutate this sequence, I can mutate back, correct? We call that a reverse mutation, or to be simple, we call it a reversion. And that happens. You imagine if I get a mutation in my hemoglobin gene that I have sickle cell anemia, if I get a mutation in that same spot, there's actually a 1 in 3 chance that I'll mutate back to wild type and I'll be normal, right? So that's a reverse mutation. Other terms to define. A point mutation. A point mutation is what you saw in the previous slide. A point mutation is a single nucleotide that gets changed. In general, we think of a point mutation being a substitution from a G to an A. Some people use the term point mutation to mean a single nucleotide that gets deleted, or a single nucleotide that gets added, and that's the incorrect way to use it. Point mutation should mean substitution. But to be clear, you ought to, as you go through your life, you ought to say it's a point mutation that's a substitution. It would be a change from G to A. Or you should say it's a point mutation in which the nucleotide was deleted. Does that make sense? You're saying the same thing twice. If you say it's a point mutation, it's a substitution, it's redundant. Some people misuse the term because they didn't learn it the way you hopefully will learn it in this class. Okay? I think word processors are a natural part of your life. You guys know what an insertion is. You guys know what a deletion is, right? Insertion is you're adding, deletion is you're losing nucleotides in the sequence. And then I want to talk about amplifications. Amplifications are a very specific type of insertion. Okay? We'll get to that in a little bit, apparently. How can mutations occur? Now, I walked through each one of these in some detail today. Most common mistakes are probably made when the genome gets replicated by DNA polymerase. DNA polymerase puts the wrong nucleotide in 1 in 1,000 base pairs. So when the fertilized egg divides, can we do this? Is that 3.2 billion? No, that's a million, isn't it? Oh, that's not. That's 3.2 billion. So when a fertilized egg divides, that fertilized egg has that many mutations in it. Yes. And then we'll talk about DNA repair on Monday. A proofreading mechanism relying on DNA methylation, which we talked about, comes in and corrects most of these. And you get down to probably that many nucleotide differences. These are point mutations, usually point mutations, meaning a substitution, one nucleotide is put in incorrectly for another one. Right? So usually DNA polymerase is making mistakes. There are other things. The environment can cause this. So this just kind of walks through the process. We talked about this before. Usually the polymerase, when it puts the wrong nucleotide in, it goes back, it pulls that wrong nucleotide out through an exonuclease activity and puts the correct nucleotide back in. So it has a proofreading mechanism. This is a second category, and this I'm going to call an amplification. And this gets back to this concept of a repetitive DNA sequence that we talked about earlier. This is amplification of a trinucleotide repeat. So here we have this sequence, ATG, that must be on the thymine, right? And we have the sequence CGG, 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 CGG. Okay, so we have a trinucleotide. We have a 3-nucleotide unit that's repeated, what is it, 5 times? 1, 2, 3, 4 times, right? That's one form of repetitive DNA. Not a big deal. You know, it codes for amino acids in here. If this is a protein, it codes for amino acids. But thymine with TTAs, and we have leucine. TCG, I don't know what that is. No idea. Phenylalanine, hydrazine, something like that. CGC, CGC, CGC. So we've just got three amino acids that are going to be repeated. Or it's one amino acid repeated 3 times, right? So when the DNA polymerase is reading along, and we got the new strand coming in like this. Where's the sequence there? So 5' to 3'. So when this puts in a C, C, G, C, C, G, what can happen is this sequence can fold up on itself. How would I do that? That would go. See how that works? Such that this thing forms a loop stem structure here. So what's happening is the DNA polymerase is reading along and adding a nucleotide, adds a nucleotide, adds a nucleotide, and adds a nucleotide, and then it's attached to the DNA that it's leaving behind. It's holding onto that. If that DNA should buckle down and pull down, it would physically, the DNA polymerase is holding on like this. It would physically strip the DNA backwards on the DNA that it's copying. And it doesn't know that it's been pulled backwards. It just says, what's the next nucleotide? Oh, here's a C, G, C, C, and it might get pulled back again. What's this? G, C, C. And every time it moves forward, this loop can get pulled longer and longer if those nucleotides hybridize through this structure like this. Does that make sense? Does anyone not picture this? This picture is weird. So we're increasing the number, we're amplifying, we're inserting that sequence, that trinucleotide repeat. And it can happen, in this case, it gets repeated 40 times before the DNA polymerase actually makes it by and is able to continue on. What will happen is the next time this cell divides, these guys will separate, so this guy goes off into another cell. This strand will be up here like this, and this is now a template strand for the next strand that comes in. We can actually draw it like that. So the next strand that comes in is going to come in and copy all that stuff, because it doesn't know. It doesn't have a map that tells it exactly what the sequence should be. It just adds on the nucleotides. So that becomes a permanently inherited part of the genome from all the cells from then on.
But wouldn't the same loop feature occur after the 40 times?
Yes. So the question is, when this thing is separated and the polymerase comes in, would this loop structure persist such that the polymerase gets stuck? Or would it maybe jump over that? Is that what you're asking?
Or wouldn't it make another larger loop structure?
Oh yeah, so you're saying wouldn't it do this? Wouldn't it copy this, and then wouldn't that fold up on itself?
Yeah, and just
yes.
become incredibly huge.
Yes, exactly. You're correct.
That's scary.
It can be. So what she's saying is, if in my body the replication increased the number of these repeats from 5 to 40, and I pass on to my kids 40 copies of this trinucleotide, in Kate and in Ellie, this 40 could maybe go up to 70. And then their kids would inherit 70 copies of the trinucleotide repeat. So let's look at the code for this protein now. We've got our methionine, what did we say this was? Leucine, I think. Tyrosine, and then CGC, is that glycine? Arginine, is it arginine? So arginine, 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 arginine. What started as 3 or 4 arginines is now 40 arginines. So in this protein, what may have started as a little loop of arginine here, now is going to develop into, oh man, what did I do to myself? It's going to be this huge loop of arginine. And it may be that this thing doesn't fold up properly. It may be that this arginine screws up the folding of it, such that the protein is not functional. Does anyone ever heard of trinucleotide repeats before? What have you heard of it in?
Huntington's disease.
Huntington's disease. There's a gene, the name of the gene is called Huntington. So there's a disease called Huntington's disease. And eventually someone was able to map where in the genome that disease originates. And they found the gene, so they named the gene Huntington. And they figured out the problem is an increase in the number of trinucleotide repeat through this process. Such that most people have 5 to 10 CGGs in the code for their Huntington protein. And every generation in humans, it gets a little bit longer. So if someone in your family if your grandparents get Huntington's disease at age 70, your parents will probably get it at age 60, and you'll probably get it at age 50. And it definitely trends that way. It becomes an earlier and earlier and earlier onset. Huntington's disease is one of these neurodegenerative disorders. I don't remember exactly what the protein is. Any questions about trinucleotide repeat? Yeah.
Could you just have like a restriction enzyme just to recognize that sequence? And
Yeah. So how do you correct it? Yeah. Do you take an enzyme that recognizes CGG and you dump that in a cell? That sequence appears a lot in the genome, doesn't it?
pretty scary.
You cut the whole genome apart. So
It's not fun.
could you make a plasmid and have a plasma that's got like 100 base pairs of this sequence and 100 base pairs of this sequence and only 5 copies of this? And the plasmid during recombination, maybe the recombination machinery would line the plasmid up with this, and you recombine this onto the plasmid and the normal wild type shorter version onto the chromosome, and then you just find a way to get rid of the plasmid. And you've now genetically modified the human back to a wild type version of the Huntington gene. That's been done in cell culture. Doing that in a human is a different matter.
A little more than one cell.
Yeah. Yeah.
Could they do that and like let's say I knew that I had Huntington's disease or whatever, and I didn't want to pass it on, so I could do like an in vitro birth and like have them do that to the
Genetically
cell.
modify your egg?
Is that like possible?
Probably. It may be a little bit illegal. You might need to leave the United States.
I'm just curiosity.
No, I mean, that's where all of this is going. So, I mean, you can actually go and have your DNA tested and they can tell you how many repeats you have at the Huntington locus. It's pretty scary because the insurance company is going to hold of that and they're like, oh, yeah, you're likely to get Huntington's when you're 50. We're not insuring you. Or, yeah, we'll give you insurance. It's $1 million a year for your insurance. Because I know you're going to get Huntington's at 50. Right? So the value of having that testing is, so I want you to imagine I get tested and I discover that I'm going to likely get Huntington's disease. My allele suggests I get Huntington's disease when I'm in my 50s. Oh man, I'm 44, and I have a 5-year-old and a 4-year-old. I think I need to start planning the next little bit of my life so that I'm leaving a wife that has a support network there. You know, I'd probably go drill wells overseas and make a shit ton of money so that she'd have a nest egg on which to raise up the kids. I would probably do that. Some people might say, well, I'm just going to quit work and I'm going to spend my last years with my kids. Because that's more important to them. You know, I've been looking more like give them a base, but different people do different
Some
choices.
people already have the base intact.
Yeah, yeah. Yeah, some people graduate from mines and have their base intact when they're 25. Anyway. Yeah. Okay. So I guess there's a YouTube video. This basically illustrates here what I drew on the board. There's a YouTube video I think that shows this. I hope this link is still valid. I didn't check it this year. All right, this is the second most common form of damage, and that is environment. Who's looking to get tan this summer? Man, when I was a kid, a nice day like this, everybody would be out. Everyone was out in the sun trying to get tan. Gonna be tan and looking good, right? And yeah, wow, I'm worried about cancer. Holy cow, my skin. Let's talk about this. So you guys know this equation, right? Or this chemical reaction here. This is a covalent bond, right? In general, when we think of a covalent bond, we think, hey, that's permanent, right? So when I draw, here's the base structure of a nucleotide. I've got, all these are covalent bonds. I've got the base hanging off here. The base is connected to the ribose sugar through a covalent bond, right? And then this is connected to a well, there's a carbon there. And then that's connected to the phosphate group through a carbon bond. Covalent bonds don't break, right? But yeah, you know that they do. So we can think of this as actually an equilibrium to this, where it's not attached. And probably the reaction is more like this. Because the odds of this thing, like, oh, let's have an equilibrium where I actually form a covalent bond back on, right? Probably doesn't happen very often. Okay, come on, so you guys know. You would talk about this reaction in terms of Ka? Yes? Biologists, we would study it for Mikhail's Menten stuff. We would use Kd, because we're opposite world apparently. You guys study association constants, and we study dissociation constants in real life. Right? So we get the Ka for this, and the number would be enormous. If it's the Kd, the number is incredibly small. How many nucleotides in the human genome? 3.2 billion. How many cells in your body? 10 trillion. So 3.2 billion times 10 trillion is a whole bunch of these covalent bonds existing in just your body. So although the odds of this happening are incredibly low, there are so many of the covalent connections like this in your body that it's relevant. And we can calculate the number of times that happens in your body every hour, and it's disturbing.
Well, I'm pretty sure that the base would not just float off. Or to get back to a happy place.
Well, it would float away, and the odds of it finding its spot again are, and if it does, how would it connect?
There's other things to help it.
No, it's
just gone floating forever.
It's gone, and you've got an empty spot in your DNA. Like a 5-year-old smiling with a big empty space right there, right? I'm getting to that point in my parenting. So, something like this actually happens. There's no purine on here, or there's no pyrimidine on there, just like floating away and it's gone. So, human body's composed of 10 trillion cells. Each hour, 1 times 10 to the 16th nucleotides need to be repaired in your body. Is that right? Whoa.
You've got to remember, billions times trillions. I don't even know where the zeros are. Somebody do that one.
That seems too big. 1 times 10 to the 13th. That's too big. That number can't be right. Because a billion times a trillion, that'd only be like 10 to the 12. Oh, but this is every hour though. I don't know. It's a lot, and it's relevant. So, these are mutations, and we'll learn next time about how these things get fixed. But basically, the machinery comes in and says, hey, there's something missing here. And so a machine comes in, it just basically takes a bite out of the DNA and misses it. So there's a single strand over here and a big space in here, and that space needs to be filled in. As that space gets filled in, sometimes mutations accumulate. Here's one. Let's compare these two nucleotides. This is cytosine, used in DNA, and this is uracil. Under the same concept that covalent bonds do actually break, every now and then this amino group will just kind of go floating away. And what you're left with is something that looks like uracil. So if the genome gets replicated, the polymerase coming along says, uracil, I don't know why that's in my DNA, but it looks like T. So opposite the uracil, I'm going to put an A. We've just changed, in the code, we've just changed a C to an A, or to a T basically in the code. Does that make sense? And it happens. You guys remember when I was talking about CPG islands? Does anyone remember what a CPG island is? You're like, ah, it's kind of familiar. Anyone?
I remember you
Travis?
saying the words GCP island.
Yeah, this is just a phosphate group. This is the genetic code. This is just the phosphate in between there. And what I talked about is DNA methylation usually happens at GC dinucleotides. That's where methylation usually occurs. And what we said is the frequency of C being followed by G is just 5% of what you would expect. And this is probably why. Lots of C's get lost through this process. If that C is important for methylation and normal gene expression, the organism would, the progeny of the organism would die off and be selected against. And so then, you keep C's in the correct spots, but otherwise the C's go away over time. Our genome is becoming less GC rich because of that. So there are a large number of these kinds of mutations where covalent bonds just kind of break. Here's another one. It's guanine. It looks like it gets oxidized. Where does that happen? Right here. This looks like something different. It looks like adenine, and so you put a T in place on the other side when DNA replication happens. So I'm not going to ask you to learn all these, but you wind up getting substitutions, right? Point mutations. Substitutions. One nucleotide for the wrong one. That's things that just naturally happen. Water, I guess, being a damaging agent. So keep away from water. Okay? Whenever you don't drink water, it's a mutagen. Next time someone tells you, oh, whatever, that's a mutagen, it causes cancer. Pesticides cause cancer. So there's water.
I'm sure it does too.
There are other things that we know of as being specific mutagens. One of these is being UV light. Remember how uptight I am in lab? You go to the UV box and I'm like, I'm putting the glasses on you this morning.
I was gonna put them on.
Yep. And I was like, I was not willing to wait 5 or 10 seconds. So I put them on her this morning. So UV light, what UV light does is if there is a thymine located right next to a thymine, the thymines form this weird cyclical structure. Covalent bonds form between these guys like this. And the nucleotides physically bend like that, and they can't be read. They can't be read by DNA polymerase, and they can't be read by RNA polymerase. And so if that's in the middle of the code for a gene, you can't use that gene until that gets repaired. Interestingly, bacteria have an enzyme called photolyase. And that photolyase recognizes this unusual structure, binds to it, and it breaks those covalent bonds. And then guess what the energy source is to break those covalent bonds? UV light. So zap the bacteria with UV light, and you will fix all the thymine dimers and create a bunch of new ones. But we'll deal with that later during the next wave of UV irradiation. Humans don't have a way of doing it. We don't have a photolyase. I wonder what would happen if someone took an egg and inserted the photolyase gene into the human genome and grew that person up, if that person would be incredibly healthy and free of cancer. I'll bet you someone's done that in mice. How about bonus point on the final exam for anyone who looks that up, sends me an email. And someone made a transgenic mouse that expresses photolyase. Yeah, if not, there you go, there's your project. It'll take you 2 years to run the experiment. Yeah, probably a year to get the mouse and a year to run the mutation experiment. But I'll bet the mice would be resistant to cancer.
At least cancer is based on.
Yeah, cancer based off UV. So that's UV irradiation. Has anyone been to the dentist recently and had an x-ray of their teeth? Like a whole jaw or head x-ray done? How many people have had an x-ray of their jaw for the dentist? Yep. So recently, let's talk about what X-ray radiation does. It breaks the sugar phosphate backbone on both strands.
Whoa. What's that?
Is your head valuable to you?
Not all the time. That's like rare, right?
Yeah. Huh?
Not rare.
Yeah. Like how many does it break?
Like how often
Depends
does
on
it
the
break
dose.
the?
Yeah, it depends on the dose. In outer space, people who spend time in outer space, this is a problem. Even though they shield the spaceships, some of that radiation gets through and damages their DNA. This one. People who live at high elevations, like a mile above sea level, have less atmosphere to absorb the cosmic radiation, and these kinds of things happen. Right? The Dead Sea's looking pretty good now, isn't it? Where is it? Where is it? Nevada? New Mexico?
The Dead
Sea. No. What's the place in Nevada that's below? Death Valley? The Dead Sea's over in the Middle East. It's below sea level, right? Yeah, the atmosphere is actually there. It's so thick that people that have problems, there are some people, machinery that repairs this. If people go and live in that area, they get relief from the lesions that they experience, that they normally experience every day. Living in Colorado would be a really bad place for those people to live that have mutations, that can't repair their DNA properly.
Can you do
So,
like a research study seeing if people at higher elevations are more prone to like cancer than lower?
I don't know of any, but that link it to this type of radiation. I think there have been studies which show that people in the mountain region get cancer more often than people at sea level, and they're comparing people at the East Coast and the West Coast, but that's kind of been attributed to all the nuclear testing that was happening in the area. And we definitely have a higher nuclear radiation background here than at other parts of it.
There's just so many like other factors that it's really kind of hard at the end of it. Okay.
I mean, you could easily run that experiment with mice. Someone may have done that with mice.
Would for something like that though, would you have to keep them in like I don't know, like the same like how would it literally be your only variable would be elevations? You'd have to have like the same humidity and everything.
Yeah,
else?
so basically you'd have like a mother mouse, maybe you have 10 mice that are inbred so much they're basically genetically identical. So you get a litter of the females, and they're all genetically identical and they're all sisters. And then you have litters from them, and some of the organisms go to sea level and some would stay up here, and you just expose them to different, leave them out so that they're exposed to outside environments so they get different cancers. don't know if you'd like it specifically to.
Yeah. Because wouldn't it be also like if they're in gold and that nuclear plant also radiation?
Rocky Flats, right?
Yeah.
They're building houses on that now, by the way.
They better be cheap.
No, they're not.
Oh, well that's stupid.
You guys know that just north of here, that Candelas that they're building? Just north of here, you take 93 north and that was Rocky Flats, and it was used to make nuclear weapons during the Cold War. And at least twice there were massive fires that released massive amounts of radioactivity that like saturated Denver atmosphere. Of course, it was top secret U.S. stuff, so they didn't tell anybody. But then when they were all done and they were shut down the plant, what they did is they had all this radioactivity and they covered up with 6 feet of dirt, so it's okay now. Okay, some chemicals look like bases, but are not bases. Has anyone compared the structure of adenine to the structure of caffeine? Has anyone compared the structure of adenine to nicotine? To see how close those chemicals look, you're really wincing. You like coffee, don't you?
I was still wincing about the Rocky Flats, but I do really like coffee.
So if you're going to drink coffee, don't buy a house in Candelas.
Okay, I'll make that sacrifice.
Okay. And especially if you've got Huntington's.
Really
Oh wait, we were way behind. So there are chemicals that just look like nucleotides, and they sometimes be incorporated into the DNA. And you get the wrong, when the next round of DNA replication comes along, DNA polymerase is like, what the hell is this thing? We're just going to put G here. And off it goes. And what it does is it says, well, I couldn't recognize that. I'm going to kick the can down the road. The DNA repair machine is going to come in and realize they don't match up, and it's going to correct it properly. See you, suckers. Yeah.
Is that what causes addiction if it doesn't get corrected?
That causes what?
Addiction? Like,
No, that's it's more with neurons.
Is that when people are always like, well, if you drink too much caffeine when you're little, you will grow? Because you won't be
No, they're
proper
probably neural
height?
stuff. They're probably neural stuff.
That might just be a threat.
Yeah.
What about kind of going along with what you said? What about like tendencies towards alcoholism?
Ooh.
Like,
I don't know. There have been genetic links to that. Certain genes predispose people to it. Like, you know, my father's father, I didn't know him too well because I was so young. My dad said he drank more than my dad thought it was appropriate, so my dad has basically avoided alcohol, and I'm making up for that.
It skips a generation.
Something like that. No, he made a purpose. He said he loves Manhattan, just like his grandfather, or his father, my grandfather. But he's just really uptight. Just make sure he's careful that I throw caution to the wind and off I go. I start the brewery. Someone else had a question? Yeah.
What kind of proteins do you make if you have caffeine in place of an egg?
Yeah, I don't know. Probably RNA polymerase would stop and be like, somebody's got to fix this crap. Or he would just say, well, I don't know, I'll just throw something in here. On I go. I think the rate, so caffeine is not a good match. It doesn't get incorporated into DNA almost never. But there are lots of compounds out there. I mean, there's a whole class of compounds that look similar. All right, we'll end on this one. Does anyone know what this compound is? Does anyone know what that compound is? Lithium bromide. So if we have our DNA, you guys saw that, cheaters. We have our DNA and our bases are hybridizing like this. What lithium bromide is this is the structure, and it's a sheet. It wants to hide away from the water, and it finds this environment here really, really nice. So the lithium bromide sticks itself in here, like this. It actually distorts the DNA around like that. If that, they call it an intercalating agent. It inserts between the bases, it intercalates into the bases like this. During DNA replication, DNA polymerase will come along and it will encounter the ethidium bromide and go, I have no idea what the hell this is. Let's put a T in here right now, and the repair machinery will take care of it later. And off it goes. So this is a really big deal. Up to now, we've been talking about point mutations, substitutions, one nucleotide for another. So you change one amino acid into a different one. Maybe you get a termination codon created sometimes. This inserts a nucleotide and changes the reading frame. That destroys the gene. Right? This is why I'm asking you to be careful when you're using ethidium bromide in lab.
Okay, so we put it in our gels in lab, so it does that to our DNA, and then when we're purifying it, you're getting rid of the agarose, but you're getting rid of that as well.
Yes, the purification process. If you have successfully PCR amplified your gene and you're now going through the process of purifying the DNA out of the gel, that process strips the ethidium out also. The amount of ethidium in there is really, really small. So it's okay. Okay, that's as far as we're getting today. Sorry to leave you depressed on a Friday. I will see you guys on Monday. We'll finish this one up on Monday.